Hi, and now what we're going to do is we're going to start this week by exploring sensation. How do we sense stimuli in the world around us? And then we're going to explore, things like seeing, hearing, and pain. Please review taste, smell on your own. Next week, we're going to explore perception and how do we understand what we sense. Now, the topic's really interesting as it helps us to understand that we don't always sense and understand the world and reality exactly the same as everyone. So now let's begin. Sensation and perception. We're going to do some quick little definitions here, okay? Now, sensation, this is the process which the senses pick up visual, auditory, and other sensory stimuli and transmit them to the brain. And then perception then is the process by which the sensory information is actually organized and interpreted by the brain. And just by the way of that graphic there, you can sort of see the differences in terms of brain, where the different parts of, walking, seeing, hearing, thinking, and remembering activates different parts of the brain. And so sensory information is entered into the brain from the different sensory entry points, if you will, and then the brain interprets it. Now this opens the door for where sometimes we don't always understand the world around us in exactly the same way. Is that maybe sensation-wise there's a deficit or an accentuation on one sense or another, and so the information going in is received differently, but also, and more importantly, will be how might the world around us be interpreted and those signals be interpreted. That has a lot to do with background and your history, where we've lived, what we've experienced, as well as the processing experience within our own brains and how that might be different from one another. So all of this can contribute to our mixed understandings, and we'll pick up on more of that in our second part of this chapter on perception. But let's carry on with the sensory world. So what is the dimmest light a lifeguard could perceive in the darkness? Now, there's a variety of things that count as absolute thresholds, measurements. These are measurements that have been established by sensory psychology. Now, just as the threshold of a doorway is dividing the entry point and the outside and the inside of the room, it divides that. The absolute threshold of sense marks the difference between not being able to perceive a stimuli and just barely being able to perceive it. Now, absolute threshold is the minimum amount of sensory stimulation that can be detected 50% of the time, whereas difference threshold is the smallest increase or decrease required to produce a difference in the sensation that is experienced or is noticed 50% of the time. And these are two types that you'll need to be aware of. For each of our senses, there are sensory receptors which detect and respond to the sensory stimuli. Now, they direct and respond to one type of sensory stimuli and convert that stimuli into the nerve impulses or neural impulses which go to our brain. Now, transduction, that's the process which the sensory receptors are converted. They convert the sensory stimulation into electrochemical neural impulses that are sent to our brain. So actually, and this is an important note, that our sense of sight isn't images going to our brain, it's electrochemical neural impulses that go to our brain that our brain interprets. Now, in all of our senses, we can establish what is known as sensory adaptation, and this is the process in which sensory receptors grow accustomed to constant and unchanging levels of stimuli over time. Smokers can become accustomed to the smell of cigarettes. My wearing shorts in the wintertime can be attributed to sensory adaptation. It's something that I've gotten used to over the 27 or 28 years I've been doing that. Daycare providers get used to the daycare environment of either being rather aromatic or noisy. And there's different sorts of, farms. I've worked on a farm, maybe you have as well, and you can get used to the smell of a farm. And these are all examples of sensory adaptation. So let's start looking at some senses and how this all takes place. And we're going to begin with the major parts of our eye and how that contributes to how we see. So we'll start with the eye, and the cornea in our experiences bend light rays inward through the pupil, and that's the small dark opening in the eye. The iris dilates and contracts and the pupil to regulate the amount of light that enters the eye. The lens changes its shape as it focuses images on objects from varying distance on the retina. And the retina is a thin membrane containing the sensory receptors for vision. Now, rods detect the level of brightness and changes in brightness, but they don't see color. And they function best in low levels lights. It's why oftentimes they are recognized to be along the edge or the side, if you will, of your eye. So if you're looking through your eyes at night, it's often better to look at whatever you're looking at from the side of your eye. So you tilt your head a bit and then look to just at the corner of your eye, and you will often see in night a little bit better because they are night receptors. They're for darkness and light. They don't respond to different colors, and they're found everywhere in the retina except the center or the fovea. Now the cones, these are more sensitive to colors, and they work best in bright lights. It's often why you'll see colors better in the daylight than you will at night. If you're wearing an orange shirt and you step into a closet, the shirt doesn't change color, but it will not look the same color as you enter into a darker area. That's because the cones need light to be most receptive. They are responsible for the sharpness of visual information, and they are found in the fovea. Now I'm going to show you something here. These are my eyes. I had my eyes tested, and I asked them if they would send me the pictures of my eye that they take. The bright objects is where the neural messages are sent towards the optic nerve. That little spot on either side of that, this little darker spot, is the fovea, and that's where the bigger light receptors are, the cones. Now when we think in terms of what does our eye see, on the bottom of this image you see a wide range of wavelengths. The invisible long waves, the invisible short waves, and they go from the AC circuit, the broadcast bands, radar, microwave, infrared, and there you see in a small little bit in the middle is the visible light spectrum, and then you got the UV or ultraviolet rays, the X-ray, gamma rays, and cosmic rays. We only see a very small segment of the entire wavelength spectrum. All right, so it's important to note that what we see in our visible eyes is quite a bit significant, it's quite small compared to what's out there. And there are other mammals and animals that see a broader range. They might even see into the infrared and the UV rays, unlike what we do. Now, if we're looking at theories that explain color vision and how that occurs, we're going to look at two. The trichromatic theory states that there are three types of cones in the retina. Each make a maximal chemical response to one of three colors, and in this case it's red, green, or blue. Each cone is sensitive to one of the colors. All colors would be perceived as a combination of these three colors. The retina does indeed have three cones. So that's one of the earlier forms or theories, if you will. Now we'll move to the second one, and this is the opponent process theory. And the opponent process theory of color perception assumes that there are four primary colors: red, green, blue, and yellow. Cones are arranged in pairs, and when one member of the pair is activated, the other is not. Now both theories have merit because each explains a different phase of color processing. Researchers believe that the color processing starts at the level of the retina, continues through the bipolar and ganglion cells, and is completed in the color detectors in the visual cortex. Trichromatic theory is consistent what happens in the cones, and the opponent process theory is consistent what happens at the ganglion cells. Now, to demonstrate the opponent process theory, try this. Stare at the dot on the green and black flag for about a minute. Then shift your gaze to what will be the blank or the white one, and see if you can see the correct colored Canadian flag. What you're seeing is, or what I hope you're seeing is, that when the green gets tired out, the green cones that are responsible for seeing green gets tired out, it loses its ability to see that. When you go to the white screen, the green is too tired to fire, and so the red shows through. So let's see if this works. Now we'll consider, hearing and the theories associated with hearing and what's involved in that. Now, sound waves enter the pinna. Now, this is the visible part of the ear predominantly, and it's kind of shaped in a way that would gather sound waves as they approached you. Now, it's the outer ear, and the sound travels to the end of the auditory canal, and it causes the eardrum to vibrate. This sets in motion the isosceles in the middle ear, which amplifies the sound, and the vibration of the oval window causes activity in the inner ear, setting in motion the fluid in the cochlea and moving the hairs, the cilia hairs in the cell receptors. This transduces the vibrations into neural impulses, and the auditory nerve carries the neural impulses to the brain. And so again, this is really interesting from the point of view that sound waves get translated into neural impulses, which are then translated to the brain. So in order to be heard, sound requires a medium through which to move. Air, water, or other solid objects can carry sound waves, and sound has characteristics that make up the details. So let's look at some of them. We'll begin with frequency. Frequency is the number of cycles completed by a sound wave in 1 second. It determines the pitch of the sound. Frequency units are measured in hertz. Now you can see that there's long and short, and all sound runs between each of these. So the shorter ones, higher intensity, the longer ones, lower intensity or lower frequency, and therefore lower sounds. Then we can move to amplitude. Amplitude is the loudness of the sound. The units of measure here are decibels. Now if we move to decibels, decibels are units of measure for loudness of sound. Now, timbre is the distinctive quality of sounds. It distinguishes it from other sounds of the same pitch and the same loudness. For example, a trumpet and a violin sound different, but when each of them play the B note. So if you play a B note in a trumpet and a B note in a violin, what you're recognizing is the difference in quality of sound, and that's called timbre. Now there's two major theories that we can consider when we talk about hearing, and they attempt to explain hearing. One is place theory and the other is frequency theory. Place theory states that the location of the hair cells in the basal membrane correspond to different pitches of sound. This explains pitch above 1,000 hertz. However, when you look at frequency theory, it states that the speed at which the basilar membranes vibrate correspond to different pitches of sound. Now this explains how the pitches below 1,000 hertz works. So frequency theory best explains how we perceive low frequencies, and place theory explains how we perceive the remaining frequencies. Now we're going to look at touch and pain. Nerve endings in the skin, the sensory receptors, these respond to different kinds of stimulation, including heat and cold, pressure, pain, and a vast range of touch sensations. The neural impulses ultimately register in the somatosensory cortex. Now, when we look at pain, we're going to look at one theory called gate control theory, which holds that the spinal signals transmitted by slow conducting fibers can be blocked at the spinal gate. First, if fast conducting fibers get their message to the gate, if fast conducting fibers get their message to the gate first, or secondly, if the brain itself inhibits their transmission. The gate control theory of pain states that when receptors sensitive to pain are stimulated, a neurotransmitter called P is released into the spinal cord, activating our pain receptors and opening up the gates in the spinal column and sending the message to the brain. Now, things like endorphins, ice, heat, and maybe even the kiss of a boo-boo will actually start to affect positively the experience of pain. That's why rubbing an area can block pain. It's what gets through the gate as opposed to what gets blocked at the gate. Now, I hope this has been helpful. We're going to pick up on perception when we get to the second part of the chapter, and we'll do that in week 4. Thank you, everybody. Keep up the good work and stay the course. Remember, time management is the biggest issue here. Use your textbook, mark it up, make notes. This is all going to help when it comes to test time. Thank you, everybody. Bye now.